Hello friends, Ace Botane's next episode. In this episode, we'll be discussing molecular basis of inheritance, fifth one. And what all things we'll be discussing in this episode. For that, I will share the screen, so it will be convenient for you. I think the screen came, so we can proceed. Molecular basis of inheritance, fifth one. And what all topics we'll be discussing in this episode? Introduction, transcription, transcription unit, template and coding strand, transcription and gene, types of RNA, and questions. As usual, 5 theory, 5 NEET model question. And these areas, these episodes, for most of the students, that topic is somewhat tougher. So it will take time to finish this chapter because subject is that much tough. So we can approach in a simple manner. So we can proceed. Introduction. So what is meant by transcription? What is its unit? And what is meant by template and coding strand? How we can differentiate it? How that name came? And how transcription genes are linked? Different types of RNAs. And 5 questions each. So first we can start with the transcription. First we should know what is meant by the term transcription. Trans, if it comes as a prefix, meaning is change, move, transfer, this way. So transcription, when we take it as a single word, this word generally means change into another form, from one form to another form. Typical example is medical transcription, law transcription, change into a readable form. Now, here in genetics, transcription means what? Here it means process of copying genetic information from one of the strand of DNA. DNA is double stranded, so one strand, that data, genetic information will be copied and that is transferred to single-stranded RNA normally. So from DNA to RNA, that procedure is called transcription. And what is the basis of it? That is a popular question. It is based on principle of complementarity. DNA duplication also, we discussed that term, principle of complementarity. And here only exception is DNA we can find adenine or adenosine. But when we discuss with the transcription that RNA will be formed, in RNA you cannot find adenosine or adenine instead of uracil. No other change, and here only single strand will be formed. So here RNA we can find uracil, DNA we can find thymine. But unlike DNA duplication, here segment of DNA will be involved, connected with one of the strand, changed into RNA. That is how these two differ, DNA duplication and transcription. I think idea clear to all of you, a popular question. Now, DNA is double stranded, RNA is single stranded. So why both the strands of DNA is not transcribed? For these different explanations are available. Most common one, connected with the NCERT, 2 points will be the one. Suppose both strands of DNA act as a template, mold. RNA molecules formed will be with a different sequence. Because 2 DNA strands are not same, they're complementary. So 2 RNA strands formed will be with a different sequence. Automatically codes for different proteins. It means a segment of DNA will code for different proteins, so the aim will be lost. That is one reason. Second reason is suppose both strands of DNA from their RNA form. So automatically both RNA strands form, that will be simultaneously, they will complement each other so that they may form a double-stranded RNA. Again, purpose will be lost. So conversion of RNA to protein will be lost. That statement is given in NCERT page number 91. I think idea clear to all of you. First page. Now we can proceed to second page. Now, transcription unit. So what is meant by the term unit? Many area we can find unit. So unit means a part. Typical example is a large area of land made up of small areas or plots or units. Now, as a continuation, it is the part of DNA which will take part in transcription, DNA to RNA, is called transcription unit. So it is the part of DNA which will take part in transcription from DNA to RNA is called transcription unit. And transcription unit has got three components. One is promoter, second is structural genes, third is terminator. So a diagram representation is shown here. Now, as we proceed, RNA will be synthesized from DNA. Enzyme involved is, RNA has to be produced from DNA. So DNA-dependent RNA polymerase. This is how that nomenclature comes. Once you get that idea, it will be easy for you. So DNA-dependent RNA polymerase. This enzyme will catalyze the polymerization, again, as in previous case, only in one direction, that is 5' to 3'. So DNA-dependent RNA polymerase, direction is 5' to 3'. And this diagram you may be observed. It is famous NCERT diagram. It is a diagram-based theory question. It is again entrance NEET type question. So it is even paragraph question. Explain the process shown here that way. So all this we'll be discussing. This is the representation, and it shows a typical transcription unit, schematic representation, NCERT. It will be explained in the next page, so we can proceed to next one. So we can continue. See, strand that has got polarity 3' to 5'. Here you can find 3' to 5' will act as template strand. Here you can find template strand. Template strand is otherwise called non-coding strand, otherwise called antisense strand. So strand that has polarity 3' to 5' will act as template strand, non-coding strand, or antisense strand. So the counter, second strand. Second strand that has polarity 5' to 3' will act as this one, will act as non-template, this one. First we discussed this one, now this is second one. Non-template or coding or sense strand, just opposite. So this is now what we discuss. The second strand that has got the polarity 5' to 3' will act as non-template or coding or sense strand. I think these two strands clear because a popular area of question. Now, interestingly, we are using the term coding strand, but remember it will not code. But then how that name came? How coding strand becomes sense strand, even though it is not coding? Reason is, it has got the same sequence like that of new RNA strand form. So coding strand is a DNA strand. So when new RNA strand is formed by the transcription, which has got same sequence like that of coding strand, except DNA we can find thymine, RNA we can find uracil. That is how the name coding strand came, sense strand came, non-template strand came. It will not code. I think idea clear to all of you. It is an important aspect. Problem type question will be there. One question I already put there, I will show it. Okay. Now, even though coding strand will not code, all references here made with the coding strand. And structural genes, that functions we'll be discussing after this. Structural genes, here you can find structural genes. They are flanked by, that is two borders, two ends. That is why I drawn this flag here. By promoter. Now, what is promoter? And terminator. What is terminator? That all we'll be discussing on both sides in a transcription unit. Now, next aspect. Promoter is located towards 5' that is otherwise called upstream of the structural gene, and is referring to polarity of the coding strand. So this area, this is the promoter. Reference is with the coding strand. Terminator, coming to this end, is located towards 3'N. See coding strand 3'N. It is called downstream of the coding strand, and that usually indicates end of the process of transcription. So the promoter position Then terminator position with reference to coding strand, upstream and downstream. That word frequently they're asking NCERT link, or I think it is clear to you. Simple only, but we should know it. What is meant by that upstream? What is meant by downstream? Promoter, terminator, what is the direction? All this. All based on coding strand, even though it will not code. Now, next is functions of 3 genes. For convenience, I separately put there. One is promoter. Promoter generally means it is a DNA sequence that provides binding site for RNA polymerase. Now, upon concept, we'll be calling it as P site. Upon the last part, we'll be studying after 1 or 2 episodes. So promoter means it is a DNA sequence that provides binding site for RNA polymerase, P site, to initiate transcription of a gene. That is how the term promoter promotes. See, presence of promoter in a transcription unit that will determine template and coding strand. So promoter is the crucial position. Strand-wise, coding strand is having a crucial position because all reference with the coding strand, even though it will not code. So by switching its position with the terminator, the definition of the template and coding strand could be reversed. Promoter, terminator, when we change, the definition of the template as well as coding strands could be reversed. That is what NCERT says. So promoter, terminator shifted. Then template and coding strands will be reversed. Now connected with this, there is a recognition sequence. That is not directly in NCERT still. Recognition sequence, that means certain short sequences within the promoter site are conserved. Now, second type of genes are called structural genes, generally concerned with the synthesis of protein or the functioning of the body. Here we can find structural genes. And third one, last part, that is terminator. Terminator is generally concerned with the end of the transcription and play an important role in stopping the process. So a starting point, body, and end point. This should be there. Many areas we can find the system. Beginning, then elongation or the procedure, then ending. So promoter, structural genes, and terminator. Coding strand, template strand. 5' 3', 3' 5'. I think idea clear to all of you. Now, next area we to proceed. I think this page is clear. Next is famous NCERT question. This is the famous one. Template strand, upper one. Coding strand, lower one. And what NCERT asks to us is, can you now write the sequence of RNA transcribed from this DNA? So from here, RNA will be formed. 3' to 5' when RNA forms, 5' to 3'. This is coding strand having thymine. Instead of thymine, you can find RNA uracil. No other change from coding strand. That is why coding strand, the name came, even though it will not code. It's a famous question, simple question. I think you got the idea how we can write it. Theory question, NEET model question. And this all are based on NCERT. Now, next is transcription unit and gene. There I written so many basic terms. Even though we heard it, many people don't know what is exact meaning. That is why I given there. See, first is cell, protoplasm, nucleus, chromosome, DNA, gene, cistron, cell. This way it proceeds. So first is cell. Here we can find. Cell we have to learn in first year. It is a structural, basic, functional, fundamental unit of life. In the cell, we can find protoplasm. What is protoplasm? It is cytoplasm plus nucleus. Protoplasm is the physical basis of life. This is a famous statement given by a scientist, Thomas Huxley. So these two over. Now comes nucleus. Nucleus is the controlling unit of a cell. Inside the nucleus, you can find chromosome. Chromosomes are supposed to be a physical basis of heredity since they contain genes. In the chromosome, we can observe DNA. DNA means basic unit of heredity. That is DNA. And in the DNA, we can find gene. So gene means it is the basic functional physical unit of heredity. And the place where genes are located on chromosome is called loci or locus. And again, last part, cistron. Cistron means functional part of gene, or we can say equivalent to coding region of gene. That is why I wrote that sequence. And various versions are available in some books, some other meaning they given. This is generally what we usually say regarding that terms like cell, protoplasm, the nucleus, chromosome, DNA, gene, cistron, etc. Various versions are available, slight variations also possible from what we discussed. Next. So we can say that gene is the functional unit of inheritance. Cistron is the functional unit of gene involved in coding. Now comes the term monocistronic and polycistronic. So we should know what is cistron. Cistron is the functional unit of gene involved in coding. So there are two terms, NCERT linked, again, exam point of view, very, very common area, important. Monocistronic and poly. Mono means single, poly means many. So monocistronic structural genes means they will synthesize only one type of polypeptide or protein. That is mostly in eukaryotes, only one type. That is monocistronic structural genes. Polycistronic structural genes synthesize different proteins or polypeptides seen in bacteria-like organisms. Upon concept, we'll be learning it. Like upon. That is the difference between monocistronic and polycistronic. Monocistronic, mostly in eukaryote organism, polycistronic, mainly in bacteria. I think idea clear to all of you. Now comes another important two terms, introns and exons. In eukaryotes, normally monocistronic structural genes we can observe, and they have interrupted coding sequence. This all diagram-wise will be coming in most probably next episode or after one more. That is why the genes in eukaryotes are split. The coding sequence or express sequence are called exons, functional one. Originally, exons are said to be those sequences that appear in mature or processed RNA. This all will be learning within next or after that episode. Exons are interrupted by introns. These introns, intervening sequence, do not appear in mature or processed RNA because they cannot code. So intron exon. This aspect is called split gene concept. Introns and exons. Exons will be functional, introns will be split gene concept. I think up to that it is clear. And this split gene concept that further complicates the definition of a gene in terms of a DNA segment, split gene. Again, making some confusion when we define the term gene or DNA, etc. And split gene concept was developed by Richard J. Roberts. You can find the figures, photograph, and Philip A. Sharp independently in 1977. And the split gene idea has got some evolutionary aspect because split gene and RNA world, that is splicing, that we'll be learning in the next episode, splicing linked with RNA world, showing importance of it. I think up to that it is clear. That is why I told you these episodes are very, very important for exam point of view theory and NEET exam. Now we have to proceed to types of RNA. Generally speaking, there are three types of RNA, major types. Ribosomal RNA, rRNA, transfer RNA, tRNA, messenger RNA or mRNA. And these three genes will be working hand to hand for protein synthesis. Mainly three types. Then some additional items are there. So first we can proceed with the rRNA, ribosomal RNA. rRNA means ribosomal RNA. It is concerned with the catalytic activity. Ribosome will be remembering catalytic RNA. Structural ribosome link, so structural RNA. And this is supposed to be most abundant RNA, 70 to 80% of RNA comes under this category. rRNA is relatively more in G and C. Size is comparatively smaller. Now comes tRNA, otherwise called transfer RNA, soluble RNA, otherwise called sRNA. It gets dissolved in the medium when they do some reactions. Otherwise called adapter RNA. That also will be learning later, structure of RNA, clover leaf shape, etc. And amount-wise, about 15% of total RNA belongs to tRNA. It is smallest. GC base pair is more. Presence of unusual bases can be seen in tRNA. That we'll be learning in clover leaf model. Next RNA for us is mRNA. mRNA means messenger RNA, template RNA, nuclear RNA, information RNA carrying information from nucleus to cytoplasm. And amount is comparatively less, about 2 to 5%. Probably longest length can vary, carries genetic information. Again, an important question, characters of rRNA, tRNA, mRNA. I think idea clear to all of you. Now, next is genomic or genetic RNA. That is exception. That is RNA act as genetic material. You already know it is seen in virus. So viral RNA representation you can find small v RNA. So here RNA will act as genetic material instead of DNA. Typical example is some plant viruses, some animal viruses, many bacteriophages. And remember, this RNA can be single-stranded or can be double-stranded. So odd RNA, mRNA, tRNA, rRNA, vRNA. vRNA is genetical RNA. I think it is clear to all of you. Now we can proceed to the next page. Here you can find 5 theory question, 5 NEET model question with the key. Hoping that it is clear to all of you. Now we can just proceed what we studied or passed through. We discussed molecular base of inheritance, 5th one. Introduction, what all things we'll be learning. Transcription, what is a unit, template coding strand, how transcription genes are linked, etc. Then different types of RNA. Then transcription, what is the procedure, double-stranded DNA, single-strand RNA, based on principle of complementarity, just like the duplication. Then in DNA, it will be thymine, in RNA, it will be uracil, only the difference. Then why both strands of DNA not transcribed? Two common reasons. One is different sequence of DNA. Second, this RNA, supposed to RNAs form, they will pair and they will proceed. Next, we pass through transcription unit. So try to identify what is unit. And this is a part of DNA which will take part in transcription. DNA to RNA is called transcription unit. 3 components, promoter, structural gene, terminator. And the enzyme is DNA-dependent RNA polymerase, because RNA has to be synthesized. That will work only in 5' to 3' direction. And this is the famous diagram from NCERT, transcription unit, promoter, structural gene, terminator, 3 components. Then structure of it, template or non-coding or antisense strand, 3' to 5'. Then 5' to 3' act will be non-template or coding or sense strand. Coding strand will not code. Then the term came all references with the coding strand due to the fact that the RNA formed will be exactly like that of coding strand, sequence, direction, except thymine uracil change. Now, functions of 3 genes. Before it, upstream and downstream. Functions of 3 genes. Promoter, that is the binding site, RNA polymerase. Then it can be changed based on position of promoter and terminator. Coding template strands may change. Then structural genes, synthesis of protein, terminator concerned with the stopping. After this, an NCERT question, what will be the mRNA form? Then transcription unit and gene, some basic terms I explained and definition given. There may be changes in certain books. Then various versions are available. After this, monocistronic and polycistronic, what is the meaning, prokaryote or eukaryote? Then we discussed about another 2 terms, intron and exon. Exon is functional, intron is non-functional, it will be removed. Then split gene concept by 2 famous scientists, that is Roberts and Sharp, independently 1977. Then types of RNA, rRNA, tRNA, mRNA, how the name came, then quantity, function, their length, etc. Then odd one, genomic or genetic RNA, viral RNA. Then 5 theory question, 5 NEET model question with the key. Hoping that it is clear to all of you. If you like this episode, subscribe all, like it, share it, comment on it. We can meet later. Take care. Thank you.